Today, we will be understanding monocytes and macrophages. Please do not get overwhelmed with this particular slide because when I'll explain all these things step by step, one by one, you will be able to understand the topic. Without any delay, let's start the topic. And this video is a part of the Innate Immunity course that I've created for all of you. The topic for today is monocytes and macrophages. Let me write it down. Monocytes and macrophages. In the last topic, we have covered the overview of immune system. Please watch that video. Now, if we talk about monocytes, they are highly phagocytic in nature. Highly phagocytic in nature. That means if you have organisms like bacteria, you have viruses, these cells, they have the ability to actually eat them alive. So they can fold membrane around these things and they'll try to eat them alive. And that makes them a part of the system called monocyte macrophage system. And this system is highly important for our protection. Monocyte macrophage system. Now let's move on to the next component is when there is this eating or engulfment of the bacteria, it creates a structure inside the cell which is also known as phagosome. This is the structure that is getting created inside the cell, and because of that, it can kill the organism inside. Now, moving on to the next one, which is now let's talk about monocytes, right? So monocytes are the mononuclear leukocytes. That means cells will have only one nucleus, and the nucleus shape at one nucleus. Let me write it down. Mononuclear, right? So mononuclear is the nucleus. If we talk about the shape, it's a usually oval shape structure, ovoid shape structure, or it can be kidney shaped structure. And the cytoplasm, let's talk about the cytoplasm. Cytoplasm will have granules, and the granules, they will be blue color, they will be gray color. When you stain them, let me write it down. They are blue colored granules, right? And the color is usually gray or blue in color. But when you stain them with basic dyes, so if you apply basic dyes and in those cells, you will see these kind of colorations. Next is origin is from the bone marrow. That is what I'm creating right now. So that you can remember from bone marrow, the origin of monocyte occurs. And then they go into the circulation. So just imagine if that's the blood vessel, the cells are inside. These are our monocytes, and then they roam around in the blood vessel in our body. So you can consider them like travelers. Monocytes are the travelers of our body, and they stay in circulation for 8 hours. After 8 hours, they move to tissue, and when they move to tissue, they mature themselves into two different cell types. One is macrophage and the second one is dendritic cell. Let me write it down. Dendritic cells. So this is how the entire story goes. You have monocytes in the blood, and when they're done with the travel and all, they will be moved to their tissues. Now let's talk about macrophages, right? And we will be detailing out all the receptors that are present on macrophages, and this becomes so very, very important for us. Let me draw the structure of monocyte, and monocyte will have a central nucleus, right? So there will be nucleus, and the size of the monocyte is macrophage is larger than the monocyte, okay? And it will have more phagocytic organelles. It will have more phagocytic organelles than the monocyte that we discussed. Next is, let's talk about nucleus that I already talked about. That nucleus is irregular in shape. And it will have a lot of phagocytic organelles, and I can label them also phagocytic organelles. And these organelles are required to perform the phagocytic. And next we talk about cell membrane. In cell membrane, it will have small structures like this. Small structures, they are called microvilli. Microvilli are present on the surface of the macrophages. And now if we they are present, they're very, very tiny, very, very small. And if we talk about, going further about the receptors, So let me draw the first receptor, which is Mannose receptor. This is the Mannose receptor. The role of this is to bind to sugar, which is mannose, present on microbes. So microbes will have mannose, and this is the receptor that will bind to the mannose. Next receptor is fucose receptor. F-U-C-O-S-E. Fucose receptor. And this receptor recognizes fucose containing carbohydrates. So basically, it's also a carbohydrate receptor. And that will detect the carbohydrate on the surface of bacteria or microbes. Next is one of my favorite, toll-like receptor. I made so many videos on toll-like receptor. It's also known as TLR in short. And the role of TLR is to bind to different molecules. Like it can bind to LPS, peptidoglycan, it can bind to zymosan, it can bind to viral nucleases, it can bind to foreign DNA molecules, RNA molecules, and there is family of toll-like receptors. Next is, this is the Scavenger receptor, and Scavenger, let me write it down, receptor. This is the receptor that binds with modified self and also microbial molecules. So that is also present on the surface. Next is, we have CD14 receptor. It's also known as co-receptor. CD14 co-receptor, or CD14, I can, let me write it down clearly. So I can write it down, co-receptor, okay? Co-receptor. Why co-receptor? Because to detect LPS, it needs to work with toll-like receptor. So in association with toll-like receptor, it will try to detect the LPS, which is present on the surface of the bacteria, gram-negative bacteria. And if we talk about some of the other receptors that are present in the macrophages, one of the most important receptor is FC receptor. And that binds to antibodies. FC receptor binds to antibodies. So let me write it down, antibodies. And antibodies, they are part of the immune system, we already know. And macrophage has the ability to bind to antibodies also. Next receptor is again very, very important receptor as the complement receptor. Complement receptor, that means it can also bind to complement. And then if complement is bound to bacteria, it can get activated. And, you know, I've talked about antibody receptors are there. So why they are there? Because these molecules, including antibodies, they can bind to the surface of these microbes. So as you can see over here, I have designed, let me change the color. So antibodies are green. Let me create those. Antibodies are green. And what they're doing is they're binding and coating the bacteria or the pathogen completely. And now this is basically where the antibodies are, or antibodies or complements are covering the bacteria. And this process, this complete process is known as opsonization. And because of this, there is high phagocytosis that happens. So antibodies actually facilitates the phagocytosis process. And this is a very, very important part of the innate immune system. These cells all together, what they can do is kill the pathogenic organisms that entered inside our body. And through opsonization, antibody-mediated phagocytosis can also happen. This is also known as ADCC. I'm not going to cover ADCC right over here because I think that is good enough for today, right? So this is how we're going to cover the next topic also. And there will be a structured flow of understanding this particular topic in very much detail. And at the same time, I'll be providing you more information on different topics related to immunology, microbiology, biotechnology, genetic engineering, so many things we're going to cover, cell biology and all, right? Stay tuned. Please subscribe to the channel if you haven't, and do share the videos with your friends and whoever you think is going to get benefited from the video. Hit the like button because that helps us a lot. Also push our content to the new audiences. Thank you so much.